So this is just a very short video to walk through some of the important takeaways about the general sense information from chapter 16. So when we think about sensory receptors, we want to begin to classify them or distinguish them based on three criteria. So the first is going to be where the receptor is located. The second is where the stimulus originates. And the third is what is the type of stimulating agent. So let's begin with classification based on where the receptor is located. So we're going to divide these into basically two major types, either general sensory receptors or special sensory receptors. Our general sensory receptors are going to be more simplistic, and we find these throughout the body. We have both visceral and somatic sensory receptors, and we've already talked a little bit, remember, the viscera, right, are our organs. They're typically made of smooth muscle. When we think about our somatic, we're thinking about motor. So we have both types of sensory throughout our body. Now, when we think about what kinds of information is being collected based on whether it's viscera or somatic. So when we think about somatic, these are going to include our tactile receptors, which we'll talk about in a minute, that are found throughout our skin. Remember, we talked about our integumentary system. We talked about different tactile receptors that detect light touch or coarse touch or light or deep pressure. So these are going to be part of our somatic sensory in our skin and in our mucous membranes. We also find somatic in our muscles, in our tendons, and in our joints. So the kinds of things that they're detecting are pressure, right? Stretch, temperature, and also pain. Now, in our visceral sensory receptors, what we're thinking about is in the walls of our organs or our viscera, also in the walls of our blood vessels. Okay, again, they're detecting stretch, right, in that smooth muscle that makes up the walls of the viscera and the walls of the blood vessels. Also pain and temperature, and even chemical changes. Now, our special sensory receptors, these are all going to be located within the head. And we're going to focus on two of these in lab. We're going to focus specifically on, hearing and equilibrium, so what's happening in the ear. And then we're going to talk about vision, what's happening in the eye. But we also have olfaction or smell and gustation or taste. Okay, now another way that we can classify our receptors is going to be where the stimulus is actually originating. So let me get my pen on here. So the first one is, we say, a receptor that detects stimuli from outside the body. So if we're talking outside, that's external, right? So those are going to be exteroreceptors. So those stimuli are coming from external sources. Now, what about from within our body? So these are going to be internal, right? So interoreceptors. And then the last one there is, these are located within our muscles, joints, and tendons, and they're specific to help us to understand our body position and our movement. These are called proprioceptors. And then our third category of how we can classify our receptors is going to be the type of agent that stimulates those. So I have here, I'm going to list them here and then we'll go through. So we have chemoreceptors. Actually, may just take them one at a time. Chemoreceptors. So what do you suppose is a stimulating agent or the modality of those? So yeah, these are chemicals, right, that stimulate these types of receptors. So what kinds of things can we imagine that would be chemical receptors? So first of all, in our blood vessels, we are detecting the presence or concentration of different chemicals. So for example, carbon dioxide levels, right? So that we can regulate our breathing based on how much carbon dioxide is present. Okay, another example that has to do with special senses are our taste buds. So as we put food in our mouth, on our tongue, we have taste buds that are sampling the chemicals present in our food. Then we have thermoreceptors. And so our stimulating agent, right, what we're detecting would be temperature or heat, right? Changes in temperature, changes in heat. So we know that we have those present in our skin, right? And if you'll recall, way back in the first or second unit, we talked about how important regulation of our body temperature was as far as our homeostasis. And so we learned that our hypothalamus, let me just write this down, is detecting, right, and regulating changes in our body temperature so that we can maintain our homeostasis. Okay, then we have photoreceptors, and we'll learn more about these in the next lesson as we talk about how our eyes are able to provide us a visual picture of what we're seeing based on the light that's coming in. So I kind of gave that away, but our stimulating agent then for photoreceptors is going to be light. So we're going to talk about that a lot more, how the eye functions. Okay, then I'm going to actually put here mechanoreceptors. And I'm going to put sort of a subheading under mechanoreceptors, and I'm going to put baroreceptors because that's sort of a type of a mechanoreceptor, but it's a specialized type. So you may have heard of a barometer, right, which measures pressure, and the weather people use that to determine what kind of weather changes, what we can expect. So the baroreceptor then is interested in pressure. Okay. So we might think about stretch of our blood vessels, right, or in the walls of our viscera or our organs. So mechanoreceptors, what is the stimulating agent? We're going to say it's distortion, right? Physical distortion, right? Mechanical, physical movement or distortion. Distortion. Okay. It could be of the plasma membrane. It could be of, as we're going to learn in this lesson, about the way our internal ear functions with these specialized hair cells and how those hairs get distorted. That is an example of that. So I'm just, for our example, I'm going to put internal ear. And we're going to learn a lot about how our equilibrium and our hearing is controlled by those specialized hair cells. And then our last one here is a nociceptor. And this is pretty complicated, but for now, what I want us to think about is these receptors are there to identify things going on in our body that we perceive as pain. So this can be perhaps the presence of very extreme temperatures or stretch that's too far, too much stretch of a particular joint, different types of things that we are going to identify as pain. So this should look familiar, right? Back when we had our integumentary system, we looked at different layers of our skin. We know that epidermis, right, is the outer layer, and the dermis then is deep to the epidermis. We also learned that the epidermis has 5 layers within the epidermis in thick skin. And then we learned that the hypodermis, or the subcutaneous layer, is that one that's deep to the dermis. Now, we also talked about some of our accessory organs or accessory structures in the skin, right? We talked about some of these receptors that detect light or deep pressure, light or coarse touch. So we're going to talk a little more about them. I'm not interested at this point in that you memorize everything about these, but we understand why we have different functions of these, and based on where they're at, deep or superficial, how that definitely determines what kind of information we're collecting with those receptors. So first of all, we have unencapsulated tactile and encapsulated. So essentially, these are going to be dendrites for the most part, right, of our sensory receptors. And so if they're not encapsulated, then essentially these dendrites don't have a covering. No tissue covering around those. So the first one we'll talk about is a free nerve ending. Okay, whoops, I meant to underline that. Free nerve ending. Okay. Now, what kinds of things would you expect a free nerve ending to detect? So, how about temperature, right? Pain. And based on where this is located, so from the image, you can probably see that that's located in this top layer of the dermis, right? The more superficial layer, which is called the papillary. Remember those papillae, right, that are formed? So that's in the papillary layer of the dermis, and they can even extend into this epidermal layer. So as you can imagine, these are going to be involved in light touch, because they're not deep in that dermis. Okay, now let's move to, go ahead and we'll cover the root hair plexus. So as it sounds, these root hair plexuses essentially form this mesh, right? They sort of form a web-like structure around our hair follicles. And as you can see, these are deeper, right? These are down into the reticular layers. Remember, we have the papillary layer, just as a review a little bit. And then we have the reticular layer, which is deep to the papillary layer. Those both make up the dermis. And so these are deeper in the reticular layer of the dermis. And these, because they're forming this web-like mesh around the hair roots, they're going to detect movement of our hairs. So an interesting thing about these is that these adapt very quickly. So we have a couple of terms that I just want to mention to you. One is tonic, and the other is phasic. So receptors can be tonic receptors, meaning that they have limited adaptation, or they can be phasic, which means that they can adapt. So I'm going to put rapidly adapt to stimulus, so to constant stimulus or stimuli. So when we think about this, so when you put on your clothes every day, right, you're not reminded constantly all day long that you're wearing clothes. And that's because like some of these, like the root hair plexus, have phasic adaptation, so they can adapt to constant stimulus. Now, the last unencapsulated receptor we're going to talk about are the tactile receptors. So as you can see, these tactile receptors, so another name that we use when we talked about the integumentary system, we called these Merkel discs. Okay, so these extend to Merkel cells in the epidermis, okay, in that deepest layer of the epidermis. So does anybody remember what that deep layer of the epidermis is called? It's stratum basale, right? It's the base layer. So they can extend into there. And so therefore, because of their location, right, it makes sense that these are going to be responsible for determining our light touch. Now let me just erase so we can start over on the encapsulated. Okay. All right. So our encapsulated receptors are going to have some kind of covering. So it could be connective tissue. It may be some of our glial cells like the neurolymphcytes, but they're not free nerve endings or free dendrites like we saw with the unencapsulated. So let's just start with our tactile, right, since it's listed at the top here. So when we talked about the integumentary system, we also called these Meissner's corpuscles. Okay, and these are going to be important in our discriminative touch. So the shape, the texture of something. These are also phasic so that they can adapt to constant pressure so that we are able to better determine texture and shape of an object as we handle it. So these are located, as you can see, in the papillary layer, right? So they're in the more superficial layer of the dermis. Now let's move to the let's look at the lamellated corpuscles. Okay, so the lamellated corpuscles are important in our deep. You can see based on where they're located, right? That these are going to be important in coarse or deep pressure. They're located in the reticular layer. They also can be found below the dermis in the subcutaneous or the hypodermis. So these are going to be important in our coarse touch, deep pressure. Okay. Now, our bulbous corpuscles, that's the last one I'm going to focus on. These also are important in determining deep pressure. These are located in the dermis, and they can also be found in our hypodermis. So I just want to reiterate, I'm not going to expect you to memorize all of these. I want you to more understand based on location that we need light touch, coarse touch, light pressure, deep pressure. We need all these different kinds of general sense, right? These are all general sensory receptors that provide us our tactile information. So we had a little review of the integumentary system, and most of this lesson we're going to be focused on hearing and equilibrium. So if your takeaway is that you can identify the different types of receptors based on these three criteria, then you'll be in great shape.